Okay, here we are again, the last session of the day. We're shifting to biology now. So the name of this talk is Big Data and Structural Computational Biology. Protein structures are defining the protein function. In order to establish what are the determinants of protein functionality, one needs first to collect and then analyze a large quantity of protein structure descriptors. In this talk, Goran will describe the process of collecting, analyzing, and deciphering data patterns in the structure-function relationship for a number of protein districts, an important approach for understanding how protein do sustain their own function and specificity in a more detailed way. So, yeah, Goran, welcome.
Thank you. Good day, everyone. So let me go directly to my presentation here. I hope you all see that. So I'm going to talk today about big data and structural computational biology. So we go from space to inner space, to biological space. And I will be introducing concepts of internal protein nano-environments, which are related to studies of how protein structural differences influence their function. When we speak about function, understanding protein function is directly related to capacity of combating diseases. Therefore, I will be speaking about some applications in medical science and designing some new agrochemicals for plant and animal protection. Two of the areas that concern any human being on this planet, basically. So when we are speaking about data, we are using structural and physical-chemical parameters. Those are our data. This is our gold mine to identify functional districts in proteins. And proteins are nanofactories present in any biological system, including human beings. So let me step back just a moment, and as you heard, my name is Goran Neshich. I'm speaking from Latin America to you right now, from Brazil, from the state of Sao Paulo, city of Campinas. And of course, you know much about Brazilian beauties, natural beauties, beaches, and so on. Those are some pictures, photographs that I took here in Brazil of wonderful avifauna. Just incredible. But you probably much more know Brazil by carnival or maybe football, maybe 20 years ago. And much less about Brazil, which is the largest producer of small and medium-sized jet planes. Even less probably about vibrant and dynamic science community. Here you see the front page of the Nature magazine, where we published first full genome of Xylella fastidiosa. This is the bacterium that attacks orange trees. And of course, Brazil is the largest producer of orange in the world. Also, we produced the Sting platform. And that platform was developed here in my laboratory. And I will be speaking about that. I work at Embrapa, where we have bioinformatics and computational biology lab. And Embrapa, according to New York Times a few years ago published, Embrapa has in 3 decades became a world research leader in tropical agriculture and is moving aggressively into areas like biotechnology and bioenergy. And that's where I work. So let's talk about Sting platform. If we consider the complete bioinformatics and computational biology platform, then we reside upon those fundamental pillars, syntactic and semantic one, and we operate over the structure descriptors and function descriptors of proteins. We were successful so much that we have several mirrors around the world, 2 in the United States, 1 in Columbia University, another one at the University of California in San Diego, where the protein data bank is residing. Protein data bank, PDB, is the data bank where we have all structures of all known proteins stored. Right now we have about 170,000 of those structures. Then we have several mirrors here in Latin America, one in Europe and one in Japan. And in 2002, you can see that PDB announced STING as an internal tool for analyzing protein structures. And what do we do here in Computational Biology Research Group? We are collecting, we are calculating, storing, analyzing, and selecting data. Data in order to decipher a mode of action of nanofactories present in all living organisms, the proteins. And our mission is to understand fully the relationship between the structure and function by collecting and mapping all respective descriptors or attributes or properties, parameters in a single database. And then make such database, which we call STING relational database, accessible for analysis through interactive interface. And that's a STING platform. And then we elaborate specific data for structure classification. And finally, we analyze protein folding, stability, function, specificity, binding, communication, etc. All of those essential for understanding, for example, how diseases become active, how we can stop them, and so on. So our research, in spite of the fact that it appears that it's data-driven, it is data-based, but it is hypothesis-driven. So our hypothesis and our major question is, what is the role of any specific protein structure descriptor or attribute or property within the constellation of all parameters that describe selected nano-environment? And the response to that is the essential to knowledge that we acquired. And knowledge we acquired, we published. And we published several papers in different high-impact journals. And you can see some of those here that I'm just listing. And those are high-impact factors. Also, STING became a tool for teaching students in some university, like here in Durham. It's part of the books. We published a couple of those. We are part of the content of some very frequented website, like this one in Weizmann Institute. And as a consequence, we organized the largest computational biology conference here in Brazil back in 2006 with the presence of 4 Nobel Prize winners. At that time, we had we've been champions in number of papers published during this conference, overwhelming usual champions like the USA and so on, which means, which shows a little bit how big is the obstacle, financial obstacle, to get to some conference outside of Brazil. Anyway, let's talk about STING modules. STING has its own web page. And then here in this slide, you see how we actually operate. Over the internet, we bring the public data. And then those public data are processed by the so-called database generator programs, which are either ours or third-party algorithms. And then we create parameters such as electrostatic potential, curvature, solvent, accessibility, secondary structure elements, contacts, dihedral angles, and so on. Once we have all these in database, what happens is that the user, through the modules which are listed on the bottom part of the screen, through the search engine, can actually acquire all these parameters and show them. And we can show them in this way, in graphical way. So here you see, for example, conservation of the sequences for the specific amino acid, alignment of the sequences, or you can see elements of the pocket. And pocket is some kind of imagination at the surface of the protein where usually drugs are sitting. And this is very important information we have to collect about amino acids that are actually creating nano-environment of this pocket. Also, we can calculate electrostatic potential at any point within the protein structure. So once we have all these elements or better descriptors, we can show them and paint them graphically as it's seen in this slide. If I read you from bottom to up, we have different kind of parameters like sponge, density, distance properties, curvature, hydrophobicity, electrostatic potential, cavities, solvent accessible area, temperature factor, relative entropy, and so on and so forth. And once we have them this way, we can actually color code them, and not only that, we can color code the intensity or numerical value for the intensity of each and any of these parameters. We can access, of course, any of these parameters through the menu of the thing, and there are different menu items that actually can lead you quite quickly to information you want. Once we have this whole database, we are able actually to make very important questions for biological problems, and that can turn out to be the real pathway toward protected intellectual property. And we were able to protect some of our findings. Of course, depends how far we went into this path. If we don't have the product, which hides the most amount of the money, we get much less. But that also reflects on how much data we have. And the more data we have, more money we can get actually in this patented intellectual property. And I will talk a little bit about that. But let me first step back once again and tell you about cells, where the proteins actually do exist and where they operate. So cell is very densely populated environment, and here is just a representation of the structure of the cell. And within the cell, we have proteins, we have membranes, we have water molecules, and thousands of proteins. So they don't exist in alone in the swimming pool. That swimming pool is very crowded one. And in that environment, proteins do fold, they perform their function, they exercise their specificity, and proteins do interact. And for all of these, there is a specific demand for particular nano-environment that has a specific function. So if we are speaking about the proteins as nanofactories or nanomachines, let me show you this machine, which is the actual macro machinery produced by the human beings. And here we can actually access cavities within that machine, like with these fiber optics. Go down there and see what's going on. We cannot do that in protein, but we can calculate. We can calculate the energy field in any point at the protein and decide how the parameters of the surrounding amino acids do influence that energy field. If we consider that there are 170,000 structure files at this moment at the PDB and protein database, which translates into 500,000 protein chains. Proteins may have more than one chain. That translates into 111 million amino acids in PDB at the moment. And for each of these amino acids we produce here in STING, 1,540 parameters. That finally translates into 40 trillion registers. That's how big is our database. That's big data. And that's an extra large database, as you can see. So once again, more data, more knowledge, but we have to have a proper hypothesis to drive our research and get to the solutions. So let me tell you a little bit about the data, more about the grouping of this data. I'm speaking about 1,540 parameters, but here we have grouped them in 97 large groups. And these large groups are further organized in even larger groups, 9 of those. So user can rather quickly find out variations of major parameter that can be measured under different circumstances and find out how the environment does behave under these conditions. And this number is always growing. So once we have this database, we can make reports. We call them Sting report, and we can do it graphically and show any of these parameters. Here in this picture, we have a collage of different windows. In the first one, you have sequence referenced parameters. Below that, geo-referenced, structure referenced parameters. And on the bottom, you have classical XY plot of the numerical values of any parameter that we can select, one of those 1,540. And then presentation in STING report is very rich. I'm not going to go into details, but what is important, like in this case, protein-DNA interactions, we can see the contacts between protein and DNA. And that's very important in the controlling the expression of gene expression and so on. So we are looking at the really molecular and atomic level of interactions between these macromolecules. We can see also how the structures are differing. Once we superpose them, you see those two proteins, blue and yellow, let's call them that way. They are very similar at the core, but a little bit different at the edges. And we can see how their parameters differ in this presentation with all kinds of parameters presented graphically. So what do we have? We are aiming actually to have all these data and parameters at the input in the so-called black box, which output is a function. That black box in our case is actually analysis, statistical analysis of main components of many of those parameters, which actually are most influential at the function. And that's what we published for a number of nano-environments. So STING applications are based on what one can achieve by using STING platform, STING interface, and a relational database. Well, we can make following type of questions. And the question we can make is, for example, how many proteins out of 170,000 that we have there are in the PDB that have a pocket volume of 35 cubic angstroms. So we pick up that specific data from our database. And they are negatively charged. And that's another data from our huge database. And 50% of that pocket are very conserved, with the conservation measured in relative entropy being lower than 20. That gives me a subset of the whole universe of proteins. That subset would be capable of interacting with certain drugs, and that's a knowledge based on data. But the hypothesis is that we do have certain reduced set of parameters that define this pocket. Okay, so our premise, as I said, is that the set of parameters can be used to describe the protein function. Therefore, we can predict the function having the structure. And here's how we do that. For example, you have three vertical white lines on the top part of this image, and those are catalytic site residues. Those residues are the residues, amino acid residues that actually perform the function of this protein. And then we can select certain parameters, and they are numerical range, to see if we can get rid of all the other amino acids and get a reduced set of parameters that defines only those three. And we can do that. And here's how we quickly get to only those three using only these three parameters. The conservation, those are very conserved amino acids. They are negatively charged, and they are at the pocket. So these are characteristics that will always, in this family of the proteins, detect catalytic site residues, those that are responsible for the function. And we can do that by selecting in many families. In 25 protein families we tested, returned positive results. So far, no family returned negative results. In this paper, we checked the usability of our data, big data. This laboratory did the experimental specific protein, DNA polymerase, very important protein in our body, including. And they made 8,000 active mutants of that protein. And they found they can change any amino acid except for one, and that one is aspartic acid number 610. And they try to understand why. But we say, why do 8,000 clones? It's good they did it, so we have experimental proof. But if you look at our graphical presentation of that position 610, and you look from the top down, fourth line, which says contact energy, you have the 610 position in orange, which is quite different of all the others. I'm not showing all the others, but just one region. That's the most energetically rich amino acid. And if you exchange it with any other one, you will cause serious consequences in the functionality. Let's talk about protein interactions. Just one bracket. One of the principal objects of theoretical research in any department of knowledge is to find the point of view from which the subject appears in its greatest simplicity. So that's our goal. Let's see how that works in, for example, interaction of two proteins. Two proteins interact over the interface. The rest of the surface is not interacting with the other protein, just the interface one. We calculate how are the parameters at that interface before and after the connecting with another protein, and we see the differences. And you can see quite clearly in these 54 selected parameters that there are some that are, most of them are not different in bound and unbound situation, but some of them are quite different. And those are the key parameters that we can use to predict where the two proteins are going to connect. Why is that important? That information is very important if you want to avoid this connection, avoid the communication of two proteins, which will suppress some kind of symptoms of the disease, for example. And with this knowledge, you can see in this picture that our algorithm for prediction of the interface is very successful, and we were the most successful one in the table where I show the data from people from Leeds, New York University, Weizmann, and so on and so forth. Just having this kind of approach as I just showed. So the conclusions are purely structural and physical-chemical information is sufficient to generate interface residue classification models with high assertion rate, high accuracy, low level of false negatives, high precision, and few false positives. Let's talk now about protein communication. This is very interesting connection between, from one side, the very basic science data-based but hypothesis-driven, and applications such as the biodiesel project. That's a very interesting area of research. And what we did, we designed something we called hydrophobic surface hydrophobicity index. We call it SHE. I'm not going to go into all the details, but I will show you that we were able to generate an algorithm that would finally be able to discern between the correct interface position and incorrect binding at the false interfaces. So if we have two proteins and they will connect at the native interface, at that position, delta She would always be positive. If we migrate the protein at the false interface, the delta She will become negative. And that's a huge advantage, having such knowledge and having such parameter based on the huge data volume that we have of pre-calculated parameters that we can use and check whether two proteins interact at the right position. For example, golden protein interacts with the green protein at that native position. If the green protein goes to the position blue or red, delta She will be negative. And that's a very interesting algorithm which we published. And that happens in basically 98% of all cases we checked, which is absolutely fantastic in biology. So protein structural differences often produce function modification. And you all probably already heard that just mutation in one single amino acid could cause death. Well, let me remind you of the famous case of amyotrophic lateral sclerosis. And there we have a protein, superoxide dismutase, which suffers very dangerous mutation at position 4 in amino acid sequence. Alanine is substituted by valine. What happens is, I show you here, one of the parameters from our list of 1,540. In this case is electrostatic potentials at the surface. You see at the left part is a position 4 where the mutation happens. But on the middle and the right part of this plot in the middle, you have you can accompany that the strong peaks in the electrostatic potential, negative peaks, have basically disappeared with the mutation. So you have the native wild type protein and the mutated protein. So it's like a Superman goggles that we have, and we can watch in different spectrums what is happening with the protein once mutation is done. And when we checked what are those amino acids that suffered electrostatic potential changes, those are exactly the ones that coordinate metal ions, which are crucial for the function. So you, once again, you have on the far left in the square where the mutation happened, and on the far right, you have in the circle where the effects of mutation happen. We call it a cat tail effect. You push the cat for the tail, but cat says meow in its head. So we don't know exactly how that happens, but we know what's happening inside there. So after understanding fully what nano-environment for folding function specificity, interfacing, drug binding is, then it's question of time to produce knowledge, and knowledge has as a mere consequence those patents. And here is one thing that I would like to actually finish my presentation. This is the patented knowledge and technology that we did. Here you see the picture of Xylella fastidiosa. This is a gram-negative bacterium, which you see in the central part of the xylem. Xylem are those big, I call it arteries in plants. Of course, plants don't have arteries, but they suck the water and solvents from the root toward the leaf. What happens is that these bacteria act like a cholesterol, and they clog the xylem, and so plant just doesn't get nutrients. So we found out that if we eliminate the movement of these bacteria, pathogenicity of these bacteria is eliminated. And what is funny is actually that this kind of protein by which this bacteria is moving is called PIL-T, is also available and used by other bacteria which attack human beings, like gonorrhea, like cholera, like bacterial meningitis. So what we did is we studied the number of algorithms and packages, among those some Stinger packages, but third-party ones and so on. So what we did, we studied the structure of this hexamer of the PIL-T. And what happened is that we found that this protein is ATPase, omnipresent, so you cannot, make changes at your will, because you can actually kill the patient. But all pathogenic ones here present in the dark on the top of this, on far left on the top, all of the organisms are pathogenic. And then below, not so dark and white, are free living organisms, those that don't create any damage to human beings. So we concentrate on those pathogenic ones, and then we look where they are very much different. Here is one which is very interesting for us. We translate that information in 3D, so we georeference it to the 3D structure of that protein, and we found that there is a pocket. In that pocket, we designed those small compounds that will be able to fit into that pocket, and that pocket is strategically placed at the interface of the 6 monomers that create the hexamer of functioning PIL-T protein. So this is inhibitor of the construction of the hexamer. And that's how we would fight this pathogen. Right now, we succeeded to synthesize one of these compounds that I show you. And we also purified the whole protein, so we can actually try experimentally to see whether our computational biology idea is actually working in the real experiment. So here is a three-dimensional picture of what we designed. And in the final moment, I will just say that we have so much data, so many information, parameters, attributes, and so on. And we, as a very careful researcher, we transfer this two-dimensional to three-dimensional data, and that's how we get to the solution. Just like in a computer, where the motherboard will receive very specific other boards with very specific function and very specific parameters. In general conclusion, we have the molecular structural computational biology is extremely instrumental in predicting events, confirming experimentally observed phenomena, helping understand biological phenomena. And also for such successful outcome, computational structural biology relies upon big data and corresponding ever-growing relational databases, as well as upon sophisticated and advanced statistical approaches for data analysis. Well, this is my small group, and if you have any questions, I'll be glad to answer them.